For those of you that don't know me, I am Sarah Fernandez-Dunn. I'm the manager of Northwestern's High Throughput Analysis Core, or NU-HTA. So if you're unfamiliar with us, we provide the instruments and expertise to automate plate-based assays with the primary mission to enable high throughput screening. So to that end, we also offer pretty extensive compound libraries for screening, including an FDA-approved set and oncology set. And so we started these user spotlights to get the word out about what we do and also give our users a chance to present their work. So today we have Dr. Craig Horbinski from the Neuropathology Division. We started working with Craig pretty much mid-pandemic in 2020, which of course was complicated, but you know, we got working together. And so his lab had developed a novel cancer model that had never been used for drug screening. And so we had a really exciting collaboration with him. And so, but I don't want to get into it. I'm going to let him tell you about his project. So with that, Craig, you can take it away.
All right. Thanks very much, Sarah. One of the projects that we're working on in the lab, excuse me, one of the projects we're working on in the lab here is treating meningiomas. And for those of you who aren't aware of what meningiomas are, they're tumors, as you might imagine, that grow out of the meninges. And the meninges are membranes that cover the brain, the central nervous system. So on the left here, you got a nice little cartoon showing how your noggin actually is layered. You've got your skin right here, and then underneath is aponeurosis. It's this dense fibrous tissue with a little bit of muscle, a little bit of skeletal muscle worked in there. Then you got your membrane that covers the bone, the skull, that's periosteum. And right underneath that is the bone itself, which is maybe about a centimeter thick thereabouts, give or take. And underneath that are the meninges. And there's three layers to the meninges. There's the dura mater, also known as, in Latin, as literally tough mother. It covers the entire brain and spinal cord. It's very, very dense fibrous tissue, very difficult to tear. Underneath that is the arachnoid, so named because it's got this gross and microscopic spidery looking appearance. It's very delicate membranes. And then the pia mater, which is a single cell layer thick, you can barely see it even under the microscope, that directly overlies the brain itself. So it's important to keep in mind that the arachnoid is where we think meningiomas grow out of. At least histologically, a lot of meningiomas, most of them look like overgrown arachnoid. And on the right, you can see an example of a meningioma, and you can see how it really is growing out of the membranes of the brain. So it's not growing out of the brain itself. Technically, it's not what we would call a brain tumor insofar as it doesn't arise in the brain parenchyma. It arises in the meninges, and that actually has some important therapeutic implications, as we'll get to in a minute. So here's what a standard run-of-the-mill meningioma looks like under the microscope at fairly high power. You get these whorls, these characteristic whorls of tissue and of tumor cells, and most of these tumors look fairly innocuous, fairly bland. But there's an incredible array of meningioma types that can occur. They don't all look like that standard meningothelial tumor. Up here is another type of tumor called fibrous meningioma, which looks more like just bland or benign spindle cells. Secretory meningioma, which secretes all kinds of goopy stuff that causes a lot more edema around the brain than you would otherwise expect for what's essentially a grade 1 tumor, a benign tumor. Then there's angiomatous meningiomas, which have lots and lots of hyalinized blood vessels, microcystic meningiomas up here. you start to get some, and these are just representative of the lower grade tumors, the grade 1 so-called benign tumors. These are tumors that mostly, if you resect them completely, probably won't grow back. But then you've got some of these higher grade tumors, clear cell, choroidoid, rhabdoid, so named because it somewhat resembles skeletal muscle. And then there's anaplastic. This is a higher power view, just to indicate the anaplasia is characterized by a lot of mitoses. And these tumors in particular can be a lot more aggressive. They can recur and behave in all sorts of unpleasant ways. For example, here's one of the things that a lot of meningiomas will do. They will not just grow on top of the brain, they will start to invade the brain itself. And on the left is a radiologic view of what this starts to look like. You get this enormous tumor that's squishing the brain, and it's eliciting a lot of radiologic changes in the brain tissue right underneath the tumor. And that's because when we look at it under higher power, you can see the tumor here, tons of cohesive tumor on the right and down here, all protruding into the underlying brain, which is here on the left. Tumors that do that are known to be substantially more aggressive than tumors that don't, in terms of their propensity towards recurrence and reinvasion. And I think you can imagine the kind of morbidity that this can trigger in patients. And not only that, but even a benign or so-called benign tumor, a low-grade tumor, grade 1 tumor, can be a real problem clinically to manage if it's in a bad location. For example, down here, this is a coronal view, so the patient's kind of facing you, looking at you. This is the sinus cavity. And right here, you've got this tumor, all this white stuff here is the tumor that's growing right in what we call the sella region, near the skull base. And these black spaces here are the middle cerebral arteries. I'm sorry, I should say the internal carotid arteries, my apology, the internal carotid arteries that are being completely encased by the tumor itself. And the internal carotid arteries are enormous, high pressure, high volume arteries that supply a good chunk of your brain. So if you're a neurosurgeon and you're trying to go in there, you're trying to resect this meningioma, you're really going to have a hard time taking care of that thing. Even if it's a grade 1 tumor, you're not going to be able to get the whole thing up, because if you try to really remove every last little bit of meningioma, you're probably going to nick one of those arteries, and then you've got yourself a real problem, because that's going to be extraordinarily difficult to control, and the patient could end up dying on the table if things go really bad. On top of that, so a lot of these tumors cannot really be resected completely just by virtue of where they arise. And then on the right here, we've got sinus cavities. So some of the tumors that arise in the brain anteriorly enough, if you look at the brain's anatomy relative to your sinus cavity, these things can actually punch into the sinus spaces. And we see this here, most of what you got is the tumor right here, but then there's actually a little bit of respiratory mucosa, the mucosa that lines the sinus cavity. So in this case, the tumor got right underneath there, and was starting to grow into the sinus cavity. Those are really, really debilitating, because, as you can imagine, it takes a lot of plastic surgery, trying to repair all that damage. And these people can really have a hard time with it. And then some of them will not just grow into the brain, they'll grow out. And this is a meningioma, anaplastic meningioma, axial scan. So the patient's front face is up here. This would actually be right. This would be left. This is the back of the head. In this case, you got this big, enormous meningioma that didn't just push into the brain, but it actually punched through the patient's skull and started growing on the outside of the skull. Now, it's not going to metastasize. It's not going to seed the lungs or any other place like that. But you can imagine a tumor that's this locally aggressive is going to cause this patient a lot of problems and could indeed be lethal. In fact, when we stratify these tumors by 3 World Health Organization grades according to increased aggressiveness. You got grade 1, grade 2, and grade 3. On the left here are Kaplan-Meier curves indicating recurrence-free survival. And then on the right, we have overall survival. And what you can see is the grade 1 tumors, yeah, even though they're considered benign, most of them are benign. We call them low grade because a subset of them will still grow back. Now, granted, it takes years for them to grow back very often, but a subset of them will still grow back. And some of them will still ultimately even be lethal. The patients may end up dying of recurring tumors that continually recur and continually invade and everything and behave more aggressively than you would otherwise expect. And as the tumor's grade up front increases, the recurrence-free and overall survivals both diminish accordingly. So even though they're not as aggressive, for example, as a typical glioblastoma, which is the most common malignant tumor that arises in the brain itself, they're still a problem. And it's interesting too, because when you compare the meningiomas with infiltrative gliomas, which is what occupies most people's attention in this field, and most research dollars go towards treating infiltrative gliomas, and rightfully so, because they're even more difficult to treat. The risk factors for both are very similar in some ways. High dose radiation can cause them. Hereditary syndromes are well known to be associated with certain kinds of gliomas and meningiomas. The older you get, the more likely you are to get one of these tumors. Infiltrative gliomas, males are more likely to get them, whereas meningiomas, females are more likely to get them. And also exposure to exogenous hormones like hormone replacement therapy can increase your risk of certain kinds of meningiomas because some of them are hormone sensitive. Whites are more likely to get infiltrative gliomas, whereas blacks are actually more likely to get meningiomas. And it's interesting because when men and or blacks get meningiomas, they disproportionately tend to be more aggressive than the ones that typically hit, for example, a 65-year-old white woman. The other thing that's interesting too is the incidence in the United States alone, roughly 20,000 on average cases per year of infiltrative glioma of all grades and all subtypes. That's what's reasonably expected in this country. Whereas with meningiomas, it's substantially higher, about 35,000 per year. Because infiltrative gliomas arise within the brain itself, they're inside the blood-brain barrier, whereas meningiomas are outside the blood-brain barrier because the blood-brain barrier only pertains to the actual brain parenchyma itself. So the membranes around the brain are not involved by the blood-brain barrier. And this is a physiological anatomical mechanism that the brain uses, the central nervous system uses to screen out or weed out and block the passage of lots of other chemicals in the body from getting into the brain and spinal cord. And it's an extraordinarily important thing. It's a very powerful adaptive mechanism to protect the brain and spinal cord. But in the case of somebody having a brain cancer, it can be a real problem because we're trying to treat these people with chemicals that are given systemically. And very often they won't cross that blood-brain barrier and get into the brain where they can do any good against the tumor cells. But for meningiomas, this is not an issue. Anything we give the patient systemically will get into a meningioma. Furthermore, infiltrative gliomas, as you might imagine by their name, have this diffuse single cell infiltration throughout the brain. It's like ants crawling through a computer. In fact, really no other kind of cancer does this, where each individual tumor cell will crawl throughout the brain. And again, that plus the blood-brain barrier makes these tumors impossible to really cure. You can't resect the whole thing. And it's hard to treat. I mean, what therapy you're going to give that's going to get at every single microscopic cell that's in the entire central nervous system. On the other hand, meningiomas, even the invasive meningioma that I just showed you, they don't do that. When they invade en masse, like cohesive tumor cells, tons of tumor cells pushing into the brain. So they're, at least in theory, easier to manage. And for all these reasons, meningiomas, should be lower hanging fruit, therapeutically speaking. In other words, we really haven't had a lot of progress in treating most infiltrative gliomas, trying to come up with chemotherapies, that sort of thing. But meningiomas should be easier to treat. Anything, again, you give them systemically, they will see it. It'll get into the tumor. Now the thing is with meningiomas, though, the research for gliomas versus meningiomas is probably at least 10 to 1, maybe 20 to 1 in favor of gliomas. And again, rightfully so, to a large extent, because a lot of people can have good results with meningiomas, whereas gliomas are almost always fatal. But even a lot of these people, like I showed you, even a lot of meningioma patients will suffer severe disease, morbidity, and either end up dying directly because of recurrent tumor invasion or indirectly by other morbidity associated with the radiation, and et cetera. And even though, we've got all this technology and all this research being devoted towards biomedical endeavors, the treatment for meningioma is still essentially the same as it was 50 years ago. For surgical resection as much as you can, followed by radiation if the tumor grows back, and maybe re-resection. And that's it. There's no chemotherapies that are standard of care or being used against meningiomas. And for years, I was taught that's because chemotherapies don't work against meningiomas. That's what I was taught. But after a while, I started getting a little curious about this and thought, well, no, wait, how many chemotherapies? And there's a lot of chemotherapies out there. How many have actually been tried against meningiomas? And the answer is not that many. When you really look at the database of clinical trials, as well as the basic science research and translational research going on with meningiomas, there's just not that much out there. So I thought, well, this seems like lower hanging fruit. Maybe we can try repurposing existing chemotherapies. And the reason for trying to go with drug repurposing is that to come up with something, some small molecule inhibitor or some other sort of therapeutic like that, and bring it into a clinical trial is a multi-year process, and that's assuming everything goes well. And everything hardly ever goes well, at least not the first time. Whereas if you're taking an existing therapeutic that is already being used to treat other kinds of disease, cancer or otherwise, and you find out, hey, we've got some data to suggest that maybe it could be effective against this other type of disease, this other type of cancer, you can repurpose that drug and skip over a whole lot of prior work that otherwise would have been necessary had this been a de novo drug. And so that was our question here. Could we repurpose existing chemotherapies against high-risk meningiomas? Again, specifically the ones that behave more aggressively, that invade, that recur, that are causing the most problems. And we were able to do this, and this is where infrastructure is so critical, because if you have robust infrastructure, you can start asking all kinds of questions that you really wouldn't be able to address otherwise. And we've got a pretty strong nervous system tumor bank here at Northwestern, where since about 2014, 2015, we've collected now over 2,500 tumors of all kinds, CNS tumors of all kinds, and matched blood, and supported a lot of active and completed research projects from it's now probably up to 35, 40 investigators, both here at Northwestern and elsewhere. This is a representative graph or figure of the kinds of tumors that we bank in the nervous system tumor bank. And the bigger the square, the more tumors we have of it. So our biggest one is going to be our IDH wild-type glioblastomas, and all these other gliomas fit in there as well. But meningiomas is a very, very large chunk of the kinds of tumors that we bank. Now, granted, most of them are grade 1, but even then, a lot of these so-called grade 1 tumors end up behaving more aggressively, and as well as the higher grades 2 and 3. And then there's metastases and all kinds of other tumors here and there. But we have a variety of tumors. Anything that the neurosurgeons take out is fair business for us to bank. So keep that in mind too, if you've got research questions, especially if you've got research questions that might involve some metastatic cancers or something a little less commonly studied. So our plan was pretty simple then. Really, it was just there wasn't a whole lot of sophistication, at least not conceptually, about this. It is just to develop a range of patient-derived meningioma cell lines. In other words, we don't just take the tumors and freeze them. We try to culture the tumor cells and get them to propagate so we can start to work with them in the lab. And then phase 2 was to take the ones that we were able to propagate and start screening them using Sarah's high throughput analysis laboratory and screening them for sensitivity to FDA-approved chemotherapies. And that's because a panel of such chemotherapies does exist in the high throughput analysis laboratory, and they've got the wherewithal to be able to do that. And then take the drugs that look the most promising in vitro and follow up on them, both in terms of preclinical translational and in vivo models, and anything that holds up there, let's initiate a clinical trial. Again, since these are FDA-approved chemotherapies, there's a huge amount of paperwork and preliminary work that we no longer need to do, so long as we can find a drug or drugs that has some potential. And again, since there are no chemotherapies to treat meningiomas that are currently standard of care, the bar is low. Anything that we find that looks good, or at least somewhat promising, can be a legitimate clinical trial. Here's the patient lines that we ended up using. Now, the top 2 lines, CH157 and Ion Lee, are very commonly used. We did not develop those lines. Those lines have actually been around a long time. They're anaplastic meningiomas, these grade 3 meningiomas. And yeah, so we just bought those commercially. But every other cell source, anything that says NU in front of it, we developed here at Northwestern. These are actual patients, their ages and their sexes are listed here. Again, like I said before, most are going to be female, and that's certainly held up here. And it was a representation of grades 1 and 2 tumors. Most were original brand new tumors, but one was a recurrent meningioma. And we set the cells, all of the cells, right up to Sarah's lab, and they use their magic to screen all of the cell lines for all of these drugs, a huge amount of drugs you can see listed here below, and ranked them by their IC50, which ones are most effective against the widest range of meningiomas. And I mean, obviously this is very difficult to look at, but what we found was when we grouped all of those different drugs by drug category, in other words, what kind of a drug are they, what kind of drug is it? One thing that stood right out was that, well, first of all, most of the drugs really didn't show what we felt were very promising IC50s. They were 10 or higher. But proteasome inhibitors consistently, regardless of the type of proteasome inhibitor, they consistently had very, very good activity as indicated by low IC50 against the broadest range of our meningiomas. Another drug class that we felt for various reasons might have some potential is also HDAC inhibitors. Some cell sources didn't respond very well, but most actually responded reasonably well. But there's a whole bunch of others here too that you can see, all kinds of different agents and pathways that are being targeted by certain drugs. Here's the top 20 drugs. So this is not looking at a drug group per se, but it's just the top 20 drugs. And you can see bortezomib is in I think it's carfilzomib. These are the 2 proteasome inhibitors. Were 2 of the top 3 out of all the hundreds of drugs that were screened. Romidepsin was another, that's an HDAC inhibitor. Panobinostat is another HDAC inhibitor. Again, these are all FDA-approved drugs. Now, there are some others that might potentially be somewhat interesting as well, but we can't test everything, obviously, to the next level. So we figured we'd start with these drugs here because they look like the most promising. Proteasome inhibitors are, bortezomib in particular, is already approved for multiple myeloma and for mantle cell lymphoma. Those kinds of therapy, those kinds of cancers are a little easier to treat because they are blood-borne cancers. So you don't have to worry as much about physical drug penetration into a solid tumor. However, at least in bortezomib's case, there does appear to be some clinical evidence of that activity against certain solid tumors. It's concerns in theory, proteasome inhibitors would, at least mechanistically, be expected to help augment radiation therapy. And that was another plus for us, where if we can add a therapeutic in conjunction with radiotherapy the patient's already going to receive, that would be very helpful. And there was at least some mixed reports about radiosensitization in these tumors. HDAC inhibitors like panobinostat, once again, approved for multiple myeloma, and like bortezomib, has at least some potential effect in xenografts in clinical trials of breast cancer and small cell lung cancer, and definitely has been reported as a radiosensitizer by multiple labs in multiple settings. So here's all the IC50s when we did it. Again, we repeated all this again in-house, in other words, in our own hands, after the screening data was produced. And we found out that yes, indeed, there's obviously going to be a range because we're talking about lots of different cell sources and lots of different IC50s. But broadly speaking, we're looking at some pretty effective IC50s in the nanomolar range. In both cases, we're looking at nanomolars of drugs activity, drug IC50, and that's encouraging, right? Because the more potent something is, even if it doesn't have perfect drug penetration into the tumor, that greatly increases the likelihood that the tumor cells will be able to see physiologically relevant doses, concentrations of the drug. And then following up specifically with bortezomib even further, this is a work in progress, so we're still looking at panobinostat as well. But we started with bortezomib, and we took two of those cell lines, the commercially available ones, CH157 and ION-LEAD, and you can see the colony formation assays. Bortezomib by itself, only 7.5 nanomolars, was sufficient to significantly drop the colony formation in vitro with CH157. Same for ION-LEAD. CH157 showed partial response to radiation. The combination was better. It's really hard to prove a synergy, in other words, specifically, at least in the context of CH157. But with ION-LEAD, there certainly did appear to be synergy, right? Because we have, although in this case we used a lower dose of bortezomib, just FYI, because the higher dose completely wiped everything out, and we really couldn't assess synergy very well. We'll probably have to do the same again with CH157. But what you see here is that the combination of these two therapeutics produces a much lower, weaker colony formation than you get with either one in isolation. And that's indicated here too by the picture of the colonies that are forming. So, all right, let's proceed further. Here we got bortezomib, and let's dose it on a daily basis for 2 weeks. And just track the body weight just to get some indication of systemic toxicity. And what we saw was compared to vehicle control, which are the blue lines, as you progress with increasing doses, daily doses of bortezomib, you get progressively, once you get past half a milligram per kilogram, you get progressively reduced body weights. So that does suggest to me a certain amount of toxicity. However, it's not that bad. None of the mice actually died, even from 2 mg per kg of bortezomib. But because that was the biggest drop, especially in this area here, we figure, all right, you know what, let's just pull back and use 1 mg per kg bortezomib, because that still should be within the range that's been used in other laboratories for solid tumors in vivo in the preclinical models. And that should still give us a reasonable therapeutic dose, especially because, again, these IC50s are in the low, they're in the nanomolar range. So 1 mg per kg should be okay. And what we saw was when we grafted CH157s in vivo, and then I waited a few days, and these are extraordinarily aggressive tumors. I mean, you'd only have to engraft 10, 20,000 cells into a mouse brain, and most of the mice, all the mice, will be dead within 3 weeks or so after engraftment. That's how fast these tumors grow, which can be a little bit of an issue because most meningiomas don't grow that fast. Even the anaplastics aren't quite that aggressive. that's another thing that's frankly held back the field of meningiomas. These are not easy tumors to model. When we take patient-derived meningiomas and try to engraft them in mice, it can take an insane amount of time for a tumor to actually form in that mouse. We've got one mouse that we engrafted 2 years ago, for example, with a higher grade meningioma from a patient. That mouse is still alive and it's got a little bump in the back of his back, and it doesn't seem to be growing any further. So that's just how hard meningiomas are to develop as patient-derived xenografts. That's why we end up using models like CH157, because they grow well. In fact, they almost grow too well. But irrespective of that, we were able to show that bortezomib as monotherapy did significantly extend median survival of these mice, at least by a few days. It was statistically significant. Radiotherapy also did. Well, we were a little surprised, a little disappointed, at least with CH157s, is that we didn't see better synergy between radiation, radiotherapy, and bortezomib. We were hoping that that would be the case, but this is still a work in progress, like I said before. We saw, at least in the colony formation, data suggesting this may be the case, but even with the ion leads, though, we may actually find, we're working on that as we speak, we may find synergy in that setting, even if we didn't really see it in the 157s. Irrespective of that, though, the fact remains that bortezomib did show some activity against an extraordinarily high-grade meningioma in vivo. And it does at least show or demonstrate that repurposing chemotherapies, currently FDA-approved chemotherapies, repurposing them against these tumors may be possible, which is something that not a lot of research groups worldwide are even exploring. So our next steps, there's a lot of things we got going on. First of all, we've got some new models of meningioma that we acquired from other investigators in this field. Hopefully more robust models that actually look and grow like meningiomas a little more realistically. And we're going to test bortezomib, and actually we're going to combine that with panobinostat, with or without panobinostat, with or without radiation. So you can see there's a certain amount of synergy we're trying to test for in vivo against not just CH157, but also ion Lee and some of these other in vivo meningioma models. And really try to work out in particular, when should you give the drug, when you give this therapeutic relative to when the patient's going to receive radiation. And if we can get some compelling preclinical data suggesting that, oh, you really should, that first dose or first few doses should be given before radiation or maybe right after, that could inform on a future clinical trial that actually could be ramped up pretty quickly once we get sufficiently robust in vivo data. And on top of that, since there is still some variability in responsiveness of these cells to bortezomib or panobinostat, it'll be worthwhile to explore both in the research set, in the experimental setting as well as the clinical setting, biomarkers of sensitivity and drug resistance. And thanks to the fact that all of these drugs are already well studied in other kinds of cancer, there's already a fair amount of literature on what kinds of resistance mechanisms one might reasonably expect when you're repurposing that drug against a new kind of cancer. So we don't have to start from square one. Well, this is my lab. In particular, the biobank technicians and Katie, the manager, have been outstanding with all this work. Shawan Ann has been a terrific research associate working on this meningioma project. I've got a number of other people who pitch in here and there. Wenja, in particular, is an outstanding research lab manager. Lots of people to thank, both past lab members, Northwestern. We have a meningioma working group within the Malnati Brain Tumor Institute that we initiated late last year. We've already had a few meetings exploring a variety of research projects, not just this one, but a lot of other research projects as well. And grateful for all the funding and support we've had over the years to move a lot of projects forward. So thanks very much. I'll take any questions if there are any.
everybody can just, I think, unmute if they want to go ahead and say their question, or you can just type it in the chat too if you want to just do that.
Hi, this is Al George. Craig, that was a really nice talk, and congratulations on what looks like a successful repurposing screen. I would like to know a little more about the assay and how that was set up and what platforms were used to do that.
Sure. Yeah, that would be a Sarah question, since what we do is we sent all the cell cultures, all of our cell sources from here downtown, and we ship them up to their high throughput laboratory up in Evanston. So Sarah, do you want to chime in on that one?
Sure. Thanks, Al, for that question. We took the cells in, we grew them up in Evanston, and then we would plate them in 384 and just do a cell titer glow from Promega assay. So it was we would do 6-point dose curves in triplicate, and then the cell titer glow just gives you your proliferation results, and we do a 3-day incubation with the drugs. So it was pretty easy, fast, straightforward assay, actually.
And that is read on which instrument?
Just a plate reader. We used, in this case, an M1000. We have the M1000 it's from Tecan, and it has a stacker, so we can do, a whole stack of plates all at once.
Just for general information, do you think the new plate reader that we're buying from Wavefront will do a similar assay?
The Wavefront absolutely can do this. It's way more than you need for it can do a whole lot more than this. It's like, using a Lamborghini to drive to school or something, but yeah, it can definitely do it.
Great, thanks.
Okay. Thanks very much. Thanks to Sarah and her outstanding laboratory for giving us the core support that we needed. I think, yeah, this is emerging as a success story. There's still a lot of ways to go, but yeah, I think we're off to a good start. Go ahead.
Craig, this is Lisa. Sorry to interrupt. I think there was somebody who directed a message towards me. What was the effectiveness of the treatments against the one recurrent tumor? This was
it was just as good. It was very good. The IC50 was, I think, it was below 5 nanomolar for that recurrent tumor. It was very, very effective. But again, that's in vitro. There's one thing to do it in vitro. We got to prove it in vivo. We're trying to grow a lot of these tumors. I didn't mention this, but a lot of the tumors that we were growing in culture, we're trying to also get them to grow as xenografts. And that's proving, like I said before, very, very difficult. That's one of the rate limiting steps of meningioma research in general, is they're not an easy tumor to model in the in vivo setting. But again, the flip side is there are no chemotherapies in use against meningiomas. So it's not like we have to prove our drug against the gold standard. There is no gold standard, at least not for chemotherapy. Even just the bortezomib research alone that we've produced, our clinicians in the meningioma working group were already very interested in starting up a multi-institutional clinical trial, because it's so simple. All you have to do is write the bortezomib script off-label, and they have that latitude to do that. So we can set up a clinical trial and monitor these people who are at risk, who have higher grade tumors and are recurrent, and they don't have any other options. Like I said, there aren't that many trials out there for people whose tumors keep regrowing and invading and spreading locally. So it's a great opportunity, really, for, strategically speaking, for Northwestern to position itself as a go-to center for people with meningiomas.
Right. And one other question from Stephanie, why did you use a different amount of drug for the IOMMLEE line than the 157 line?
Yeah, that's because it worked too well. 7.5 nanomolars wiped out all the tumor cells. So it's hard to test for synergy when the drug killed 100% of the tumor cells. And that's the trick with the dosing. In fact, that might be a reason why we didn't see better synergy with CH-157 is we're constantly trying to finesse the dosing, especially relative to the amount of radiation we're giving these cells in vitro and in vivo to really get a handle on what we might reasonably expect from this combination, this therapy combination in patients. That's where the devil's in the details.
So it looks like that's it on the chat questions.
Thanks,
Right.
Lisa, for reading those questions out to us. And thanks, Craig. That was really great. We really appreciate you taking the time to do this user spotlight with us.
All right. Thanks, everyone.
Bye, everyone.